Yo, what is up YouTube? Thanks for clicking on this video. So if you've been around my channel at all, you'll know that I'm a fan of education and I like to learn. Recently, I have decided to apply for a graduate certificate in biological data science. And I come from a programming background, not a biology background. So I'm going to need to bridge the gaps there a little bit. And one way to do that, I've heard, is through this platform Rosalind. Now this is mainly like a programming platform that's meant to teach you like the basics of Python for bioinformatics, which, to be fair, I do know Python pretty well. I haven't done Python for bioinformatics specifically, but I imagine it's the same principles. Really what I want to do is start to learn a little bit of the biology as I go through this and then tie it back into the programming. So I found that this is the perfect platform to do just that. So we're going to get started with the first problem, counting DNA nucleotides. And what you'll see about all these problems is that they have a expansion where it gives you the biological context. So for this, I'm gonna break to a little short video, go through, summarize what this says, and then we'll get into the code. So let's do that.
Every living organism is made of cells. Inside most eukaryotic cells is a nucleus, basically the cell's command center, and packed inside the nucleus is a tangled stew of material called chromatin. During cell division, also known as mitosis, that chromatin tightens up into chromosomes.
Chromosomes are built from nucleic acids, long chains called polymers. Each link in the chain is a nucleotide, a sugar, a phosphate, and one of four bases, A, C, G, or T. This sequence of bases is what makes one DNA strand different from another. DNA is nature's storage format. Nearly every organism on Earth, from bacteria to humans, encodes its entire genome using these four letters. In this problem, Rosalind gives us a DNA strand and asks one of the most fundamental analytic questions in bioinformatics. How many A's, C's, G's, and T's does it contain? It sounds simple, but this is the foundation of nearly every genomic pipeline.
Okay, now let's get into the code. So I've already set up my environment here in VS Code, and I've used a virtual environment in case there's any packages we have to install. I've also created this little utils.py class. Since the problems come in as a text file, all this is going to do is allow us to store our files here in this data folder. And then we can call this text file to stir method that I just wrote, which will take the text file and then return it as a string. I use this strip just in case there's any trailing white space that you want to get rid of. And then when we're done solving the problem, we want to write our answer. Usually that's a string that we can just write to a file. So then I have this answers directory that I've created here, and all this will do is we pass in our answer, and then that'll just write it to the answers directory for us. So I am gonna download the data set here, and then let's move it into our data folder. Okay, so we have an example length of 21 DNA string. Of course, the alphabet is A, T, G, and C. And we want to return 4 integers separated by spaces, counting the respective number of times that the symbol A, C, G, and T occur in S, respectively. Okay, so this is, in my opinion, a perfect opportunity to use the counter class. And that is from the collections library. So anytime you have a problem where you see return a count of a certain number of elements in an iterable, in this case the iterable object we have is a string, you should immediately think counter class. So how can we do that? We do from collections import counter, and I'm also going to import our utils class, create a util object. Okay, And then first thing we're going to need to do is get our data, which we have here. So this is just our string of nucleobases. So that is, we can easily get that. We'll just call it DNA is equal to util dot text file to string, and then the name of the file is Rosalind DNA.text. And then all we need to do for the counter class is we just assign a counter object to the DNA string we just created, because this is an iterable object. So for that, we would just do, we'll just call it DNA counts maybe is equal to the counter of DNA. And if you want to see what this actually returns us, let me just print it off real quick. And then I'll run it here with Python problem. So I don't know why my screen just did that, but you can see we have a counter with 254 A's, 234 T's, 212 T's, and 204 C's. So what order did they say they wanted us to return it in? A, C, G, and T. So for that, we can just simply do right answer, util.right answer, and then we do an F string of DNA counts of, let's see, A, and then we do a space. And then they said A, sorry, I gotta look at it again. Oops. I gotta look at the order again. A, C, G, and T. Okay, so then we have, I'm just gonna copy this 4 times. So then we have C, G, T. And then if we run that, we should see an answer. Oops, I did not pass in a file name. I'm just going to call this one.text because this is problem 1. Okay, and then we write it, and then there is our result. We're going to just copy that. Let's copy the answer here. And there we go. And that is the first problem. So this has been the first episode of the Rosalind Bioinformatics Stronghold Challenge. In the next episode, we will get into transcribing DNA into RNA. Thanks for watching, and I'll see you in the next one. Bye-bye.